Hi, my name is Melissa, and I am a second-year PhD student in the Department of Cellular Biology, and I am honored to be considered as a finalist for the ARCS scholarship. Humans are made of small units called cells, and if I asked you to picture in your mind what a cell looks like, you might imagine something like this. A cell with organelles, or specialized structures, such as the mitochondrion, that are separate and distinct from one another. The inside of each organelle is unique from all of the other parts of the cell, but what is it that keeps the insides from just floating away? The answer is that organelles are bound by membranes made of molecules called lipids. Each organelle requires a continuous supply of lipids, but how do these lipids reach the organelle membranes? For years, scientists have thought that molecules travel to organelles via simple diffusion or by trafficking in specialized compartments called vesicles. But now we understand that organelles are actually packed all throughout the cell, and they can interact with one another directly at specialized areas of contact called membrane contact sites. In humans, membrane contact sites are very important for lipid transport between organelles. A class of proteins that transports lipids at membrane contact sites is called start domain-containing proteins. Start domains can bind to lipids, extract them from the membrane of one organelle, and deposit them into the membrane of another organelle. In humans, plants, and animals, we know a lot about lipid transport at membrane contact sites, but we know much less about lipid transport in other divergent organisms. I study lipid transport in a class of organisms called apicomplexans. The apicomplexan parasite that I study is called Toxoplasma gondii. Toxoplasma possesses an organelle called the apicoplast that has 4 lipid membranes. The apicoplast is important because it is a target for drug development against the parasite. Toxoplasma is sometimes called the world's most successful parasite. It infects an estimated 1/3 of the population, causes devastating disease, and can infect all warm-blooded animals. My work with Toxoplasma addresses how the 4 membranes of the apicoplast are supplied with lipids. I study a start domain-containing protein called TG-start that localizes to the apicoplast. This protein is similar to a protein in humans called STARD-E7 that localizes to the mitochondrion and supplies it with lipids. When TG-start is present, the apicoplast remains intact, but when TG-start is depleted, the apicoplast loses its integrity and the parasites die. I hypothesize that TG-start maintains the membranes of the apicoplast by transporting lipids from another organelle at a membrane contact site. My future work will go into understanding what organelle TG-start mediates a membrane contact site with and what types of lipids it binds to. These are important questions to answer because not only will they shed light on how other organisms maintain their organelle membranes, but it could even open the door to potential therapeutic approaches against this parasite. Thank you.